All right, imagine trying to build a ridiculously complex machine, but you have a blindfold on. Well, that is essentially the core challenge of manufacturing at the nanoscale. We're manipulating matter at the level of individual atoms and molecules to create these incredible new materials, but we can't exactly use a standard optical microscope to just look at what we're making. So when you're engineering something literally 1,000 times smaller than a single human hair, how do you actually know your chemical recipe worked?
Right. How do we map the invisible? How do we prove beyond a shadow of a doubt that a specific molecule successfully attached to a nanoparticle, or that a complex chemical reaction completed perfectly? Well, we use a highly specialized tool called FTIR, which stands for Fourier Transform Infrared Spectroscopy. In the fast-paced world of nanotechnology, FTIR is basically the ultimate detective's magnifying glass. It allows researchers to identify, confirm, characterize, and map chemical structures at the nanoscale, all without ever needing to physically see them in the traditional sense.
Okay, let's dive right into this explainer. Here is our roadmap for today. We'll start by looking at the nanotech detective. Then we'll break down how FTIR works, explore green synthesis, dive into functionalization and composites, check out advanced nFITR, and wrap up with why FTIR matters.
Starting with Section 1, the nanotech detective. So when you're working with microscopic materials, you obviously can't rely on visual cues like shape or color. Instead, you have to read chemical fingerprints. Every single molecule vibrates in a very specific, unique way when exposed to infrared light. Reading those precise vibrational signatures is exactly how we identify what we're working with down at the nanoscale. Moving right along to Section 2, how FTIR works. To really understand how we read these chemical fingerprints, we need to look under the hood and break down the mechanics of the actual machine. The whole process relies on this incredibly brilliant, high-tech scanning sequence. It all starts with an IR source, which fires off a beam of infrared light. This light travels directly into an interferometer, which uses a clever system of moving mirrors to split and then recombine the light beam. This creates a highly complex interference pattern. This manipulated light then hits our sample. Let's just say it's a cluster of gold nanoparticles. Now, the molecules in that sample will absorb specific wavelengths of the light based entirely on their chemical bonds. Whatever light isn't absorbed simply passes straight through to the detector, which records the final signal.
But here's the catch. What actually hits that detector is a raw, squiggly wave called an interferogram. To the human eye, it just looks like a completely messy jumble of noise. And this is exactly where the Fourier transform part of the name comes in. Fourier transform is essentially a complex mathematical process that acts as a translator. It takes that raw, messy interferogram and magically converts it into a clean, highly readable FTIR spectrum graph. On this graph, we track wave number along the bottom axis and transmittance up the side, which lets us read the specific peaks and valleys that make up our chemical fingerprint. So by reading that spectrum graph, researchers aren't just taking wild guesses. They get precise functional group identification and incredibly detailed chemical bonding data. It reveals exactly how different molecules are interacting with one another. And honestly, the best part? It does all of this extremely rapidly, and it's totally non-destructive. That means you don't have to destroy your precious, expensive nanoscale sample just to analyze it.
All right, Section 3, green synthesis and stabilization. Let's look at a real-world application of this. Traditional nanoparticle manufacturing often relies on some pretty harsh, toxic chemicals. But researchers are increasingly pivoting to a green approach, combining natural plant extracts with metal salt solutions to synthesize nanoparticles in a much more eco-friendly way. But how do we actually know that the plant extract did its job and created the nanoparticles? While the researcher compares the FTIR spectra from before and after the reduction process, they're basically looking for the disappearance or the shifting of specific functional groups like OH or C-double bond O peaks between the two states. When they see that shift, it's the definitive proof. It's the smoking gun that confirms the reduction of metal ions down into zero-valent nanoparticles.
And it actually does double duty here. During this green synthesis, FTIR also identifies what we call capping agents. These are the phytochemicals, proteins, or other biomolecules from the plant extract that literally wrap around the newly formed nanoparticles. They stabilize them and prevent them from just clumping together into a useless mass. The FTIR spectrum proves that these protective biological layers are present and actively doing their job. Let's move on to Section 4, functionalization and nanocomposites. This is where we look at how we modify these particles and mix them into much larger materials.
Now, a plain nanoparticle? Sure, that's useful. But a functionalized nanoparticle? That is an absolute game changer.
We can actually customize these particles by attaching very specific molecules right to their surface. We're talking polymers, Y-shaped antibodies, powerful drugs, or targeting ligands. Doing this allows the nanoparticle to, for example, actively seek out specific disease cells in the human body.
And FTIR is the ultimate quality control inspector that confirms these functionalization successes. It verifies that the covalent or non-covalent binding of those ligands actually happened by detecting their unique chemical signatures right there on the particle surface. Plus, it assesses purity. It meticulously scans for any residual surfactants or unreacted chemical precursors that might be left over from the manufacturing process. If there's a stray chemical hiding in your batch, FTIR is absolutely going to spot its specific peak on the spectrum.
Now, when we scale up and start building larger nanocomposites, which is basically mixing these nanofillers into a polymer matrix, FTIR does two critical things. First, it evaluates if there's proper chemical bonding between the nanofillers and the surrounding material. Second, it monitors dispersion. It tracks structural shifts in the spectral peaks to tell us if the nanoparticles are evenly dispersed throughout the composite, rather than clumped up in one corner, which would totally ruin the material's structural integrity.
Section 5. Advanced Nano-FTIR. Because as amazing as standard FTIR is, science literally never stops pushing the boundaries. Let's look at the cutting-edge evolution of this tech. You see, conventional FTIR has a physical limit based on the wavelength of light itself. It's called a diffraction limit, and it severely restricts just how small of an area it can actually analyze. Well, Nano-FTIR completely shatters this barrier. It combines standard FTIR with a technique called scattering-type scanning near-field optical microscopy, or s-SNOM for short. Essentially, it uses a microscopic metallic needle to focus the infrared light down to an impossibly tiny point, which enables hyper-localized chemical analysis of nanoscale materials.
And how tiny of a point are we talking about here? Nano-FTIR achieves a sub-diffraction spatial resolution of 10 to 20 nanometers. Let that sink in. That is absolutely mind-blowing. We are talking about the ability to analyze the chemical makeup of materials at a scale just slightly larger than a single strand of DNA.
With this kind of incredible resolution, Nano-FTIR allows for literal chemical mapping at the nanoscale. Researchers aren't just looking at a single average graph anymore. They're getting highly detailed topography images right alongside hyperspecific chemical maps. They can look at individual nanoparticles or nanowires and pinpoint exactly where the carbon-hydrogen bonds are, where the carbon-oxygen double bonds are, and where the oxygen-hydrogen bonds live, right down to the nanometer.
Which brings us to our final part, Section 6, Why FTIR Matters. Let's just zoom out to the big picture for a second. Why does all this highly technical analysis actually matter for the real world? It matters because the vital attributes of FTIR, the fact that it's rapid, it's relatively simple, non-destructive, requires minimal sample prep, and is just incredibly versatile, make it an absolute engine for modern research and industry. It is the exact tool that ensures drug delivery nanoparticles are perfectly engineered for biomedicine. It helps us design massively more efficient components for sensors and energy storage. It verifies the nanomaterials used to clean up pollution and environmental remediation. And it directly guides the development of smaller, faster nanoelectronics.
FTIR isn't just a machine sitting in a lab. It's the fundamental bridge between theoretical chemistry and functional, real-world nanotechnology. It gives us the power to build with absolute confidence in a realm we cannot even see. And as we continue to push the boundaries of science, with resolutions now reaching down to an astonishing 10 nanometers, we really have to ask, what invisible worlds and what life-changing materials will the FTIR detective map next? Thanks so much for joining me on this explainer. I hope you found it as fascinating as I do. Keep asking questions, and we'll catch you next time.